Good morning, ladies and gentlemen. Welcome to GenScript webinar, Circular RNA Synthesis and Lipid Nanoparticle Delivery, a comprehensive solution for enhanced RNA therapeutics. The approval of mRNA-based COVID-19 vaccines marked a significant scientific milestone, establishing mRNA as a promising drug modality. However, linear mRNA faces challenges due to their short half-life and immunogenicity, limiting their broad applications. Today, we promise to take you on a journey into the exciting world of circular RNAs and their revolutionary applications to overcome these limitations, guided by the expertise of our distinguished speaker, Dr. Kevin Chen. Dr. Kevin Chen is the Director of mRNA and Plasmid Departments at GenScript and a prominent figure in the field of genetic medicine. Dr. Chen holds PhD in microbiology from South Dakota State University and did his postdoctoral training in veterinary science at Kansas State University, where he studied the type 3 secretion system in EHEC and EPEC. Since joining GenScript at the end of 2016, Dr. Chen has been at the forefront of developing plasmid production and mRNA synthesis, and he is going to bring a wealth of knowledge and expertise to our discussions. Before we get started, here are a few housekeeping notes. We have a dedicated Q&A session at the end of this webinar, so feel free to leave your question at the Q&A box and we will address to them later. This webinar is being recorded and the recording will be made available to all participants afterwards. And now, without further ado, I'll pass the stage to Dr. Chen. Dr. Chen, over to you. Dr. Chen, you need to unmute yourself.
Can you hear me?
Yes, perfect. Thank you.
Okay. Thank you, Bonhua, for the introduction. And it's really an honor to share this work to all of our friends and customers. So today, I'm going to introduce the one-stop solution for circular RNA and LNP deliveries. That's some work that I did in GenScript. So my talk will cover three different parts. The first one is the IVT mRNA introduction, and the second one is circular RNA and LNP delivery. And the third part is a GenScript CircRNA and LNP platform introduction. So mRNA is transcribed in nucleus and it is exported to the cytoplasm. So in cytoplasm, they have two different phases. The first one is it will be recognized by the ribosome, the translation complex. And this translation complex will form kind of a loop to start the translation. And there's a protein called a poly A binding protein, which will bind into the poly track and drag it close to the translation complex to form the loops. In that way, the translation will start. And here is an electronic picture shows the translation process. Here you can see this black beads are ribosome complex. And this line, this is the mRNA template. And this branch sense is the nascent protein that has been translated. Alternatively, there's another fate for the mRNA. As we know, the half-life of mRNA is quite short. So there's multiple enzymes in the cytoplasm that can recognize the mRNA and degrade it. So, for example, the adenylation enzymes, so it will eat the poly track and degrade the mRNA from the 3' end. Also, there's another different enzyme which called a DCP2 will recognize the cap structure and start the degradation from the 5' end. So eventually, the mRNA will be completely degraded. And here is a slide to show the mRNA technology development. So around the 1960s, scientists found the mRNA, and later scientists used the in vitro transcription method to produce the mRNA. And but they found that the mRNA can cause some cellular immunogenicity issues, which is a big barrier for the mRNA to be developed as a drug. So in 2005 and 2008, scientists like Karlik and Wiseman from University of Pennsylvania, they found that modification to the uridine can bring down the cellular immunogenicity and improve the RNA stability and the translation. And they were rewarded as a Nobel Prize this year. And later, there's many mRNA drugs was developed. And in 2019, the outbreak of COVID-19, and which make mRNA vaccine is a superstar in the scientist world. And mRNA vaccine is a very effective strategy to fight against the COVID-19. So here is a slide to show the basic components of the mRNA. And the first one is the file capping structure. The ribosome need to recognize the file cap structure and start the translation. So the cap structure is very important for the protein expression level. And the second one is the untranslation regions. The sequence in this region, they form a very unique secondary structure and to recruit the ribosome and improve the protein translation level. And the third part is the coding sequence. For the coding sequence, scientists can do like a modification such as codon optimization to improve protein translation, or do modification to the uridines to bring down the cellular immunogenicity. And the last part is the polytail. Polytail is very important for translation and also to protect RNA and improve the RNA stabilities. And here is a slide to show the mRNA capping process. So in the IVT process, we mainly use the T7 iron polymerase to do the IVT. So the T7 iron polymerase binding to the T7 promoter, and usually the TATA box, after TATA box, it's the start nucleotide of the transcription. So the IVT use the negative strain in the DNA template to start the transcription. So for example, if the negative strain is the first nucleotide in the negative strain is the C, the first nucleotide transcribed is a G. You can see the RNA transcript here is stuff from the G. And here is the uncapped RNA structure. So you can see the naked triphosphate at the file end. And here is a capped RNA structure. If there's a guanine linked to the triphosphate here, then it's a cap 0 structure. If there is an actual methyl group added into the first nucleotide here, and then it's a cap 1 structure. If there's another methyl group added to the second nucleotide, then it's a cap 2 structure. And there's two different strategies to add the cap structure to the RNA products. The first one is enzymatic reactions. So basically, you can add two different enzymes into the IVT reactions, like guanine transferase and methyl transferase. So the first enzyme add the guanine to the file end, and then the methyl transferase methylate this rings here. Alternatively, there's there's a co-transcription method to add the cap structure to the RNA. Basically, you put the cap reagents into these IVT reactions, and eventually you will get capped MI products. And here is some picture shows the capping efficiency between different cap method. The ARCA is the first generation of cap reagents developed by Trinic. So it's a cap zero structure, and you can see the capping efficiency is about 70 to 80%. And here is the enzymatic capping RNA, and you can see the capping efficiency around 80 to 90% efficiency. And here is the co-transcript capping structure. You can see the capping structure efficiency is about is more than 90%. So majority, most people choose the co-transcriptional way to add the cap structure to the RNA. And here is a slide to show the MI base modifications. This is the Y type of nucleotide in DNA and the RNA. And here is like the modified nucleotide in the RNA. This is a Y type uridine structure, and this is the pseudo-uridine structure. Basically, there's a nitrogen replace the carbon in this range here. So this is the only modification in the pseudo-uridine. And there is another type of modification which is called N1-methyl pseudo-uridine. It's add another methyl group to the nitrogen here. And we have done many internal tests. The N1 methyl pseudo-uridine is a little bit better than the pseudo-uridine. So that's the default modification that we recommended to our customers. Also, there's some other type of modification such as N5C. So the function of this modification is to slow down the MI degradation and also to bring down the cellular immunogenicity. And this slide should show the different strategy to add a poly track in the mRNA. And there's three different ways. The first way is to use the poly-A polymerase to add the poly-A tails in the mRNA. Because this is an enzymatic reaction process. So the number of the poly-A in the final product, it will be it's a wider range. And the second way is to put the poly-A tail in the plasmid DNA template. So and then linearize the plasmid DNA template. So eventually do the IVT, and you will get a MI product with the poly-A tails. And the last method is to put the poly-A in the PCR primer and do the PCR reactions and use these PCR products to get MI with poly-A tails. Because this is it's based on PCR template, so so it's quite difficult to scale up, and the cost is quite high if you want a large quantity of MI. And there's different strategy to design the poly-A. So typically you can like design 100 A's in the poly-A tails. This is most people will do. Alternatively, because poly-A tail is very unstable in E. coli, E. coli does not like this kind of poly sequence, so it will intend to delete or lose some A's in the sequence. So scientists from biotech, they found that if you put a 10 nucleotide space between like the first 31A and the second 71As, and it will help a lot to stabilize the poly A tails in the E. coli cells, and to save the cost and the turnaround time for the plasmid generations in E. coli. So the second part, I will introduce the CircRNA and MPD delivery. So CircRNA has been found many years ago, and originally they were found to function as like mRNA sponge that can bind into some RNAs and silence the gene functions. And also it can function as like a protein sponges. The idea is the same, it's binding to the protein and prevent its biological functional. And but recently scientists found that the CircRNA can also translation protein based on the sequence in the CircRNA. So it's kind of like a very popular field in the science world now, that scientists want to use synthetic CircRNA as kind of like a drug for the disease. So how to make a CircRNA in vitro? There's a different way to make CircRNA in vitro. The most popular way is to use the type 1 intro self-splicing method. So here is like picture show the process, and we can use the intron sequence from different species. So design some homology arms at the 5' end and the 3' end. Eventually, these two homology arms will form a kind of like a binding to each other together, and bring this intron sequence together to form the unique secondary structure. And at these regions, it will start a nucleophilic attack to the nucleotide, and eventually it will form a CircRNA. This is the basic process for the self-splicing method. Alternatively, you can use T4 RNA ligase to ligate the linear RNA and to form the CircRNA. And so we know that for linear mRNA, it's the translation is a cap-dependent ribosome entron mechanism. So the ribosome recognizes the 5' cap and form the loop and start the translation. For CircRNA, it does not have a 5' cap structure. So scientists design IRES sequence at the upstream of the start codon. The IRES was originally found in a virus. So the virus used this region to recruit the ribosome and start the protein expression. So we put the IRES sequence in the upstream of the start codon and turn on the gene expression. And here's a slide to show the difference between a linear mRNA and a CircRNA. So the linear mRNA usually has to design the polytrack in plasmid. And as I mentioned in the previous slide, E. coli does not like the poly A sequence. It's quite difficult to prepare the plasmids. And then for the 5' cap structure, as I mentioned, we usually use the co-transcriptional way to add the 5-cap structure to the RNA. And TriLink has patterns to protect itself in these regions. And also the URD modifications, which I mentioned in the previous slides, this also has some pattern limit. And delivery, such as LNP, there's many patterns for the LNP. And for CircRNA, we don't have to design a polytrack in the sequence. So, and also the circularization method was developed by GenScript. So there's no IP limit for RU products. And here I'm going to introduce the LNP delivery. So LNP is a lipid nanoparticle, and it contains different lipid compounds, such as ionizable lipid and cholesterol and nuclear, and also the mRNA and some help lipid and the PAC lipid, which to stabilize the LNP structure. So as we know, the nucleic acid is a negative charge. So the ionizable lipid is a positive charge, has some positive charge. So it can bind to each other very closely. And then the cholesterol and the help lipid to fill into the space in this structure. And some PAC lipid will add into the structure, stabilize the whole LNP. And here is an LNP encapsulation process. So the different lipid compounds was dissolved in ethanol, and the mRNA was dissolved in like PBS buffer, and then mixed together in the microfluidic machines, and we got LNP raw material, and then we would do buffer exchange to change the buffer, and then we would do some QC tests, such as the encapsulation efficiency, to check how many mRNA was packed in the LNP, and also to check the LNP size, and also the zeta potential to check the quantity of the mRNA. So there's many different applications for CircRNAs, such as vaccines, and also gene therapies, and CAR-T therapies, and protein replacement therapies, and genome editing. So the large part, I will introduce the GenScript CircRNA and the LNP platform. So CircRNA strategy was founded like about 10 years ago. Daniel Anderson from Harvard University, they found that they can use the type 1 intro sequence from Anabina and the T4 phage to do the circularization. And in GenScript, we also developed our own circularization method. So we compare the circularization efficiency of these two methods. So the length 1 and 2 is the sample used the method developed in the paper. And this is the precursor RNA, which is like a linear format for RNA. And this is a CircRNA. And you can see after like a circularization, our method has a much higher ratio of a CircRNA in the samples. You can see the ratio here. And we use Agilent 5200 CE machine to check the purity of the crude samples in this experiment. you can see our crude sample has a 75% purity, and the method described in the paper is about 52% purity. And then we use HPLC method to purify the CircRNA. And you can see that there's different fraction collected in this process. The highest fraction is about 90% purity. And under the same condition, the highest fraction of this of the method described in the paper is about 80%. So our method, circularization method, has a higher circularization efficiency. And this is the mRNA-LNP production workflow. So first we design the template and synthesize the DNA sequence. And then we do the cloning and plasmid preparations, and then cut open the plasmid and do the IVT reactions, and also the purification. And then we do the quality control and submit it to the LNP encapsulation process. And here is the half-life data for the CircRNA in vitro cell assay. And we use enhanced EGFP as a reporter gene. And this is a linear version of EGFP. You can see the fluorescence signal decreased after like 3 or 4 days later. And in around 1 week later, the fluorescence turned to dark a lot. And the CircEGFP, you can see in the first day, the signal is not very strong, but after 3 days later, or even 1 week, it has a very strong fluorescent signal here. And we try to quantify the data, and you can see that the pattern is like the linear mRNA, linear EGFP reaches its peak at day 2, and then the protein expression level decreased a lot after 1 week. And for CircRNA, it's reached its protein expression peak around day 6, and then it's slowly going down. After 12 days, still have a lot of fluorescent signals. And we also want to check the cellular immunogenicity in vitro cell assays. So we check different protein expression, such as IL-6, interferon alpha, and NF-kappa P expression. So we have different treatment. The first sample is an uncapped mRNA, and the second one is phosphatase treated uncapped mRNA, because the uncapped RNA has triphosphate groups at the 5 end, and it's believed to cause the cellular immunogenicity. So we use phosphatase to remove these groups, and then the Cap1 mRNA, and also we use phosphatase to treat this sample and to see its cellular immunogenicity. And the fifth sample is CircRNA, and these two samples are negative controls. So the transfection reagents only, or the cell only. So you can see that the uncapped and Cap1 structure, it still have some cellular immunogenicity, and the phosphatase treated sample and the CircRNA has much lower immunogenicity. And the same pattern was observed for interferon alpha and also NF-kappa B. So the conclusion is like the purified CircRNA has lower immunogenicity. And we also did in vivo animal study. Like we because transmembrane protein are quite difficult to be expressed in mammalian cells, and it's also very challenging to produce antibody for this type of transmembrane antigen. So here we try to use CircRNA as a method to produce antibody for transmembrane protein. We choose the CCR9 transmembrane protein and make the CircRNA, which is about 90% of purity, and packed in the LNP. Here we use the Modala SM102 lipid formula in our LNP. And here's some LNP QC data, like the size is less than 100 nanometers, and the PD1 is less than 0.2. And here is a encapsulation efficiency is about 90%. And here is a concentration of the mRNA that encapsulated in the LNP. So we produce this mRNA and encapsulated in the LNP, and then inject into the mouse, and we immunize the mouse every 2 weeks, and then we collect the serums after the second immunizations. So you can see after 1 week of the second immunization, there's not too much antibody detected in the serums. So basically we have 5 different mouse in this study, 6 mouse in this study, and here's a positive control, and this is the negative control. And then at the second week, we we test the serum again. You can see that we detect very strong antibody signal in the effects for all these 6 mouse. So the conclusion is that CircRNA can be used as a way to for transmembrane antibody productions in the animal. So that is our animal study case. And here is some this is a summary for the QC and its specs. And for the CircRNA, we will do some QC test to such as the identification, purity, and impurity, and also safety. For the LNP, we also provide some concentration, capping encapsulation efficiency, and also the nanoparticle size, and the PDI test, and the zeta potential test. And all this test, we provided 2 different QC package. The first one is like the RUO standard QC package, but some projects require higher QC standards. So we also provide upgrade options for the QC test. So for example, the CircRNA identification, we can use RNase R to degrade the CircRNA, and if it cannot be degraded by the RNase R, it's proof that it's a CircRNA. And also, like we can check the purity by HPLC, and also the impurities such as the protein residues, and also the DNA template residues, something like that. So that's pretty much for all the information that I want to share to you today. And I'm very glad to take any question from you. And yeah, that's all. Thank you.
Hey, hi, thanks, Dr. Kevin. for the great presentations. Yes, we do have some questions here. For all the participants, if you have any questions, please feel free to type it in the Q&A box. So the first question is, what are the advantages and disadvantages of self-splicing intron and also T4 RNA ligations for the circular RNA synthesis?
Oh, okay. Thank you, Bong Huang. This is a good question. So, oops. So the self-splicing is more efficiently, and also it's easy because it's only at a binding at a GTP to the reaction, and then it will automatically do the self-splicing. For the T4 RNA ligation, you have to add another. You have to add an enzyme T4 RNA ligase into this reaction, and the cost will be higher. And also the T4 RNA ligation process, sometimes it can ligate two RNA together instead of one CircRNA. So it has a more different byproducts. So that's the disadvantage of a T4 RNA ligation process. So in GenScript, we use the type 1 self-splicing method to do the circularization. Thank you.
Okay. Meanwhile, Dr. Kevin, you can also check the Q&A box because I'm going to read out the questions. So just for your reference. The second question is it able to produce scarless circular RNA?
That's a good question. Yes. Scarless CircRNA is a very good direction, and I know that many company and lab is doing some work in this field. And yes, we have in GenScript we have developed our own scarless CircRNA, and we are going to launch this service probably next year. Yeah, in 2024. Yeah.
Okay. So do circular RNA require modified nucleosides?
That's a good question. No, because maybe, but in our internal test, we compare the modified CircRNA and unmodified CircRNA. We didn't see big difference. So by default, we did not do modifications, but it won't hurt the protein expression. So it really depends.
Okay. And customer is, our attendee is also asking that, hang on, there's too many questions. I'm flooded by all the questions. Okay, so how the circular RNA can be used for peptides or peptide-based drug development, and how efficient it will be in commercial level?
That's a good question. So peptide-based drug development, as far as I know, that there's a technology called mi-display technology, which can be used to screen peptide or antibody, I guess. So I worked with someone that focus on these regions, and pretty much they use this kind of method to screen peptide and use peptide as a drug. So that's the thing that I can think of. Yeah. Thank you.
Okay. The next question is, okay, so just now you talk about linear mRNA and circular mRNA, right? So how do we differentiate linear and circular RNA that has the same length? Is there any way to differentiate them?
Oh, yes, because usually we use a specialized agar gel that purchased from Thermo, and you can run the agar gel to separate the linear mRNA and the CircRNA, because CircRNA they kind of like form a circle format. So and also there is a few nucleotide byproduct that was removed from in the process. So there's, I would say, like 20 or 50 nucleotide difference. But I guess the question is like, if you have exactly the same length of a linear and a CircRNA, can you see the difference in the agar gel? Maybe, but that would be quite a difficulty to see a big difference. So yeah.
Okay, let me read out the next questions. What is the longest circular RNA length produced and the purity level for that?
That's a good question. So because of CircRNA, the longer of the sequence, the higher chance you will get a nicked CircRNA byproducts. So usually we can do length less than 3 kb and has a good circularization efficiency and purities. If the sequence length is larger than 3 kb, that will be quite a challenge. Yeah.
Okay, and I think I can ask these two questions together. So is it easy to purify circular RNA? And um, what is the maximum quantity of circular RNA for each purification run?
Well, so for CircRNA, it really depends on how you want to use it. So, for example, if you want to use a CircRNA as a kind of like antigen to put like a vaccine, and the impurity will not be a big issue. But if you want to use like CircRNA as to express a protein, such as like a CAR protein, then the impurity will affect your experiment data. So just like you can use the lethal chloride to do the and RNR to do the purification, which can get quite a good purity. But at the same time, you can use a column chromatography like HPLC. We use a reverse phase HPLC to do large quantity CircRNA, and it has very good purity. So it really depends on the downstream applications and also the
Okay.
quantity you want. Yeah.
Okay, perfect. And so our customer is asking, do you have any experience regarding an antibody delivery by circular RNA RMP?
Yes, I shared one case study in today's talk, and yes, we use the CircRNA to express the transmembrane antigen and try to make antibody in the mouse, and our data show very good result. Pretty much in this case study, we have like more than 10 clones generated in the mouse, and it all has good EC50 data. Yeah.
Yeah, and the next question is, how many genes can it effectively translate on one construct? And is there any size limit to the total mRNA?
How many genes? Well, if you talking about the linear mRNA, the length is more flexible. Like you can get as long as like 9 or 10 kb. But if you are talking about the CircRNA, as I mentioned in the previous question, the maximum length we can do is 3 kb right now. Yeah. So you can do the math. How many, for example, if one gene is 1 kb, then you can do like 3 genes in the CircRNA. Yeah.
Yes. And do you have GMP service I think he's asking about mRNA.
Yeah, we do have a GMP service, but provided by ProBio, which is a subgroup in GenScript. So our team is mainly focused on non-GMP servers. So, but if you have a GMP service, non-GMP and GMP service request, you can contact us, and we can do internal tech transfer to the GMP group. Yeah.
Okay, and the next question. So customer is wondering what LNP size is for antibody delivery and how to route the administration um, could affect a half-life in vivo.
Can you? I didn't find the question. Can you repeat again?
Okay, so customer was wondering what LNP size is for antibody delivery.
The size usually the LNP size is about less than 100 nanometers. So the smaller the better. So but our maximum size is 200 nanometers. So that's a very low quantity. So the best quantity is around 100 nanometers. So like 80 to 120 or 150, the maximum. Yeah.
Yes, and how to make sure the circular RNA is circular one is produced? Can it be linearized again accidentally?
I didn't get the question. Can you see that again?
Oh, okay. So sorry. So how do we make sure that when a circular RNA is synthesized, will it be linearized again?
Oh, so like after we make the CircRNA, how
Yes.
can we make sure it will not be nicked again?
Yes.
We would check the purity of the CircRNA using the antigen, the CU 5200. So if there's a nicked products in the samples, we can see a different peak in the final data. So that's how we can check the quality of the CircRNA.
Okay. I guess we will answer one more question, and for the rest of the questions, we will answer it, after the webinar. I mean, we will answer it in emails. So the last thing is, what is GenScript circularization's efficiency?
That's a good question. So it's the CircRNA is the circularization efficiency. It's a little bit depends on the whole sequence, because sometimes RNA can automatically form some secondary structure. So in our internal data, the circularization efficiency can be as high as 90%. We have many cases of we can reach to 90%, but sometimes we can also get as low as like 50 or 60% circularization efficiency. So it really depends on the coding sequence or the whole RNA sequence designed.
Okay, thank you, Dr. Kevin. So let's take in one more question. I think it's follow the question just now, asking the LNP size for antibody delivery. So he's asking how a route of LNP administration could affect a half-life in vivo.
That's a good question. So it really depends on the downstream application. So, for example, if you want to make antibody production, so you can inject like the MP on the skin. That's quite a typical way to do that. But if you want to deliver the mRNA into the blood, most of the time, people inject the LNP through the tail blood vessel. So, that's inject the LNP into the tail blood vessel is very tricky. Sometimes the mouse will die if your LNP quality is not very good. For example, the size is too large, and it can cause some blood clot and cause the issue to the heart, and eventually mouse will die. So if you inject the LNP on the skin, that's it's more like flexible, so the mouse is more tolerant to this kind of administration way. So it really depends on the downstream applications. Yeah.
Okay, we still have a lot more questions, but I think we'll answer it, after the webinar. We will share all the answers and all the questions and answers with all the participants afterwards, so you guys can have a look. So do check out your emails from us. And for those who request the slides for these presentations, I will check with Dr. Kevin, and maybe to make a version for customer. So I think this is the end of this webinar. Thank you so much, everyone, for joining us, and have a nice day. Goodbye.